Okay, let's start. We have now 1.5 hours for preparing the summaries and conclusions from the breakout session. And yesterday, I think we started a bit with the other groups. But perhaps I pass now to the rapporteurs and how you see the situation.
No, Andrew was just asking about Professor Shankar Narayanan. I don't know how many of you are familiar with his work. He is a classical geneticist, used to work in Leiden. He passed away on May 28th, and he used to be with, Unscare, was involved in many reports. Yeah, many reports, and ICRP as well, and he just passed away in Leiden on 28th. I knew before coming here, but during travel, I totally got confused. Yesterday, I received the information that his funeral will be tomorrow. I forwarded you the thing, and I can forward you. People can offer condolences on the funeral home website, and probably because we are dealing with genetics, maybe we can just 1 minute silence in honor of Professor Shankar Narayanan. Yeah. I mean, I was writing to Nori earlier about this, and he also familiar with his work, and it's classical genetics, Dobzhansky evolutionary genetics and this type of work. I think I used to interact with him because I spent 3 years in Leiden with Professor Natarajan. So we used to bump into each other, and I used to take help from him in writing manuscripts and things. He's a very good writer, and he wrote many Unscare reports since 60s. Yeah, 60s, 17, yeah, radiation genetics.
Indeed, he was the mastermind, and also the previous recommendations, they are based on his work, so that's a loss.
So I think for the summary for what we've done, I don't think we've come to any large overarching conclusions, but rather we've been identifying issues that could be priorities or maybe directions for further prioritization. If there's any conclusions, and I think that the general feeling is that in epigenetics, it's a promising field, but there's very little information available just yet that contributes to a quantitative risk estimate in relation to those in future generations. So there's a lot of interesting material coming out, but nothing that can easily be translated into risk estimates. Whereas for the more the genetic or heritable effect in offspring, there's slightly more. So like the updated ABOM survivor data give a weak indication for effects on malformations, on pregnancy outcomes, not so much on germ cells. So it's mostly the malformations and the pregnancy outcomes. There we consistently see an effect. It's weak, and the confidence intervals are large. I think they fail to reach significance, but because it's so consistent, it's not so clear if we can disregard that. And there is a bit more quantitative information. So there is information about risk per gray. So there is a difference for the epigenetics and the more classical effects. I think that's if there's any conclusions that we've reached, then it's probably that. And then there's a lot of questions we've been considering. And in the progress since 2007, the trio studies are interesting, but the dosimetry often is problematic. So for those, they provide a lot of fundamental insights. But again, for risk estimates, at the moment, we can't really look to those. It could be interesting to see if we could establish more, because for the ones we have now, they're all being run long after exposure. Like for particularly the British veterans, they're now all so old that by selecting the ones that are still alive and have not had cancer, there's a huge selection in there. So if you could do this in a population where the exposure was more recent, you might have less of that effect. So I think that's for the and another new development since 2007, which perhaps we all got so used to that we don't really realize anymore that it was new at the time, is that these genome-wide studies were just becoming available at the time. So there is a lot of information at the genome level. And for example, in the TRIO studies, we can probably expect to see more there. It also means that when we're transferring information from animal models to humans, rather than just looking at single locations in the genome, it might be more informative to look at the whole genome. Because as Cisco pointed out, if you look at the mice appear to be more sensitive, but it could be the choice of genes considered rather than a global effect. So that would also be a recommendation. So that's the things I looked at last night. I have more in my notes, but I think this is the main things that came up when I looked over them in the evening. And there's a lot more, there's the things that we discussed with group C for the experimental models, where I think generally the conclusion is that it's hard to make a global recommendation because it all depends very much on the endpoint you're looking at. But I'd have to look at that in a bit more detail. So I think these are sort of the main things, and then there are a lot more point-wise things. So I had a look at what I wrote down for the epidemiology and took some notes for that. We can go over what I wrote down to see if that's in the right direction, and if you think so, then I just continue doing that for the other things.
Can I just add something? That was a really good summary. I just wanted to highlight again, I mentioned it yesterday, that we started a systematic review that I think it was about 2017. Now, whether we've got all the search criteria that people are interested in, whether we've made it too broad, but the point is that we've done it. I brought the human data here that we've been collating, and we've got, so it's split into human and animal studies. We're probably, and then we've split it into environmental exposures, which is the largest group, and it's actually a really difficult group to be able to put together. So we've got less collation of, or organization of that data, but the occupational and exposures are clearer. And we've then split them into pregnancy outcomes, variety of different pregnancy outcomes, and done meta-analysis on a few of the studies where we can. That's what I want to update with the ABOM studies and any other studies that we've not captured in the past couple of years. And then we also have cancer, non-cancer, and genomic anomalies. So so there's a lot there, and actually we want to be able to put this together, and maybe with some help as well for sections. But what I'm offering is that some of it might be useful for summaries.
Are they future generations or in the exposed?
No, it's all future generations. Yeah. But we haven't captured papers in the past couple of years. But I think once sections of this will be parts, I think we can see it as different reviews because they're too big. So certainly they can maybe contribute to some of the summaries. Given what you said yesterday, that the 2007 report was publications up to 2000, then I think we'll be capturing more in this.
I made a note. So there's, I think, one thing that we also need to discuss is how much detail do we want to go into in the summary presentation? Because there's a lot in my notes. Like, for example, which ages should we be looking at? And there, I think our answer was it depends a lot on the endpoint. Not everything is obvious at birth. Like, as Cisco pointed out, there's the very serious malformations. 2 or 3% background you would see at birth, but a lot only become apparent later. So which age you should look at depends on the endpoint you're interested in. It's not always obvious that you'll see everything at birth. But is that the sort of thing we want to discuss, or do we think, well, is anyone in the audience going to take that home, or is it something that should be in our notes and that we can discuss when we're writing our review?
I think that we can do both, actually. So have the global summary where are the main points, and then go into more detail, which is important for the ICRP as well. I think you had a very good global summary, perhaps of the epigenetic effects. So just to recognize that it is a kind of new mechanism for heritable transmission potential, but it's probably more important in the somatic cells than in the germline, and we don't indeed currently know the impact for the future generations. And in fact, I have the feeling that they are kind of incorporated already in the health outcome. So the question might rather be which is the genetic and which is the epigenetic contribution in the health outcome. Are there any effects that could be then more, that are not considered now but could be epigenetic? The multifactorial, they haven't been really measured as a health outcome in genetic studies, hereditary studies, at least.
That we also discussed that, especially in the field of epigenetics, we can also take use of looking outside radiation science and taking results from findings not radiation related, because these processes are probably similar.
Yes, I think yesterday the general feeling was that epigenetics certainly is interesting, but before we start trying to understand how they work in future generations, it's probably good to study them in more detail in somatic cells. And that doesn't mean they're not relevant for future generations, but from the point of risk estimates, we're not ready yet.
And of course, then I think that we have the global summary, then go to more details, and then we can indeed create recommendations for future research, what is needed. Obviously, the whole genome approaches are very important, and they should be continued both in the human populations, the TRIO studies, and have additional information from animals like the mice to get an estimate of mouse mutation rate. That would be helpful.
That it is important to use the suitable methods to detect really changes characteristic for radiation. He gave this example of indels, which is not necessarily detected by NGS, because using largely applied fashionable techniques might lead to wrong conclusions just due to the wrong approach.
So would it be a good idea to go over our recommendations together now? So I think the details I have, and I can make a selection of those, and maybe I'll miss a few things in the presentation, but they can always come back in the review. But for the recommendations, it's probably good if it's something that we think of together. Because the summary, I think I have, but the recommendations might be good to go over those again.
So, okay, if we can, how do you like to work? Do you want to prepare something now and then we can look into that, the text or presentation together, or?
For the recommendations, I would like to come up with them right now together, so they're shared, and then I can write them down, and we could always go over them again together. Or if there's no time, you'll just have to trust me to more or less get it right, but I can check with you. But for the summary, I can't guarantee that there won't be something I missed, but you'll all be in the room, so you can always interrupt if there's something you feel should have been there that isn't, and that, I think that probably is the best way to go. So I'm trying to capture as much as I can without making it too long and without throwing too much information at everyone. But if you think I've missed something important, then just add it.
So just to add to the the whole genome or the trio study type work, is thinking about different exposure scenarios, so internal and external, and also for the animal studies, similar, but to think about dose, because the animal studies that's been done, is that not 3 gray, I think? I think it's a higher dose. So to think about dose for the animal trio studies.
As you now you mentioned dose, there should definitely be a recommendation to have good dosimetry as well. So variation in dose, variation in exposure, and dosimetry. Yeah. Oh, yeah. Yeah. So dose rate, chronic versus acute, internal, external, yeah. And radiation quality. Yeah, yeah.
And to be sure that the deletions will be analyzed. So because this is for radiation quite critical. The deletions from the methodological point of view. Yes, we have Svetlana, please.
Nice to see you again. I have a general comment for the report. Perhaps we should differentiate it. The effects of acute and long-term preconception exposure for the offspring, and also focus at the differences in the duration of preconception radiation exposure. What do you think about it?
Did you catch? Please repeat. Yes, we didn't quite hear.
I mean, we should differentiate the effects of acute and long-term preconception exposure. Between
Yes.
we will see any differences in the outcomes in offspring. And maybe this question is very important for the report.
Yes, thank you.
I like the summary. Couple of comments, suggestions. When you talk about the epigenetic knowledge, I think you said little knowledge, maybe little and or complicated to draw clear conclusions about the relevance. And another one, should we also discuss endpoints of current literature? Like we know genetic mutations, epigenetics was said, and what about others? Maybe we can draw some information from your preliminary results, because you mentioned cancer incidence. I don't know if you have some other disease-related. Is it possible from your current state to say that there is evidence of this type of detriment or disease occurrence, or it's preliminary? It's not, you're not in a position to to say that, okay, we see genetic effects and we see also cancer.
So so for the cancer, there's definitely, so the non-solid cancer, there seems to be evidence, but again, this might not be, I'm trying to, I don't have in my head if they're the most recent papers or whether they go back to some of the Sellafield. They also will include some of the studies from, when was that? The 1990s. So, but the outcomes that we've collected on are all pregnancy outcomes. So that includes congenital, that includes sex ratio, birth weight. There's stillbirth, neonate death. So there's different categories under all pregnancy outcome. And then there's solid cancers, non-solid cancers, and then the other kind of collects a few things that just don't fit into any of those categories. And then we have a category on genomic, genomic anomalies. So in other words, that would be whether there's sort of samples taken and then non-epidemiological health outcomes. So, chromosome aberrations or mutations, that sort of thing. So we've pulled quite a lot together, but there's still a few things I want to pull out that maybe shouldn't be in there. But and then we can discuss, we can look at them all in a little bit more detail.
Yes, thank you, Rona. And another point, I don't know if we should discuss or anticipate this. I know that the last group, ABC D group, will be asking some questions about the mutation, the doubling dose for mutation, which is like for calculation of risks. Are we going into this direction or no? Just in case, or we just wait until they ask and see, because it's one thing you were touching upon that on the one, science, like what's scientific knowledge there, and then there's radiation protection. So I'm sure they're discussing this from the point of radiation protection, the formula. Is there knowledge to update this number in the formula or not?
Just as an intermezzo, I just realized that one thing we should probably have in our recommendations as well, because we talked about that, was that we recommend to consider all outcomes, including those with uncertain relevance for detriment, because they may actually turn out to be relevant later. I think that should be one of our recommendations too. As to the formula, if you look at that, you implicitly assume that the dependence of the effect on the dose is linear, and given a very little, certainly for epigenetic studies, with the very limited quantity of information we have, I'm not convinced that will be the case. So it is the simplest assumption you can make. So perhaps it is, I'm not saying it's not valid, but we don't know. So if we're using that formula, you assume that there is a linear dose effect relationship. And yes. No, I said it's not. No, so I think, I don't think we know this.
This is what comes out from the animal studies, yeah, of course, that's very clear. Yeah, but also from the megamouse experiments. This was the first data that came out, and that was very clear. And there was a very clear dose rate effect as well. And I think the question is, what they all want to know is whether the value is still valid, and that is something we don't know. And we just don't know. But that's one of the things that should be looked into and to test.
Would that be something, do we recommend to go back to the literature to do additional tests?
I mean, another thing that, because you can think about this further on and say, well, we have been assuming until recently that there are no transgenerational effects in humans. So we consider them in the system of radiological protection just to be on the safe side. So the first question is, are there at all? And then I think whether the doubling dose, what is the value for this? That will come later, I think. So maybe we shouldn't jump too far. But then And then again, this value of doubling comes from animal experiments, and I don't know whether there are any new data. I'm not sure if people are looking at this. Maybe from this Institute of Environmental Sciences in Japan. I don't know. I don't know. But they're looking at cancer, not at mutations. So again, I don't know.
I agree. I agree that actually we need to collate and summarize all where we are with evidence that there is weak evidence or there's clearer evidence for an actual effect, and then we can take the next step. So I completely agree with Audrey.
Yeah, because just to come back to this, I mean, we have to remember that the data that we saw yesterday or the day before yesterday from Hiroshima and Nagasaki, there are no new data. This is just reanalysis of what has been done before. So you have the same data set, but it was just reanalyzed, and booms, here you go. You get those response, which is not significant, but there is a trend.
Yes, and with respect to the last year's study from Chernobyl human liquidators group, they haven't seen an increase in de novo mutations, but they notice a trend in mother's exposure can have some effect. So maybe because it's a substantial study, one of the most robust studies so far with whole genome analysis, sequencing analysis. Maybe we should recommend to, I don't know if they're doing that, I'm not familiar with the authors and their planning. Maybe more should be like, we should look more into the effects in mother's exposure based on their conclusions and convert that to animal studies.
Because I mean, when preparing for the presentation, I really studied very carefully this paper on trios on offspring born to Chernobyl liquidators and evacuees. And for maternal exposure, the only thing that's been shown in association between the telomere length that we also discussed and maternal gonadal dose. Overall de novo mutations, the spectrum of de novo mutations has no association with maternal dose. So that was the only parameter, telomere length, that was significantly p.03 associated with maternal gonadal dose. So I would not say that we should look more specifically on maternal than paternal, because it's not evident exactly. And in terms of the atomic bomb survivors data, it's reprisal, but also Svetlana presented on the congenital malformations in the Mayak workers cohorts. That's another piece of information that goes to the bulk of evidence or our knowledge. And just to speak a little bit about the recommendations, we were asked many times, what is our conclusions about how many generations we should go? Shall we put something like that in recommendations, or it should be in more specific things or overall recommendations for the human population to go at least for 3 generations? Shall we highlight that, or? Yeah, I don't know if it's smaller or it really should be also,
When we said 3 generations, so the exposed parent, the child, and the grandchild, that was rather specific for the prenatal exposure, where you don't have the issue that the fetus may be exposed, including germ cells already present. So if you're looking at prenatal exposure, we think 3 is optimal because it captures a lot of information, and it's still reasonably practicable to consider 3 generations of humans, whereas with 4 it already becomes very hard. But I don't know if that's still true if you're looking at chronic exposure that continues throughout pregnancy.
Could be the case in case of in the scenario of protracted exposure, but what we saw yesterday, I think it's a very, I would say, specific example, because we have various combinations of the exposure of parents, and it can be only father exposed, only mother exposed, both. So I would be, not following this provocative path, saying that we should revise the definition of what is F1, F2, generations. No, that was really specific case, and I agree that was not the more provocative than.
Perhaps it is good to have some general recommendations for human studies, such as consider both the mothers and the fathers, aim for 3 generations. Do we want to have that in our conclusions? Did you know that actually include mothers and fathers? It may not sound very spectacular, but it actually probably is important.
Apologies. So one thing about doing this study is that it's incredibly difficult about the timing of conception. And I think that, looking at just paternal exposures, you're very clear. So
Yes, that was the point, to discriminate between early before conception and longer times before conception, and that's relevant to biological germ cell development stages, sensitivities. I think that's important. I agree from biological standard. Yeah.
Well, there are not many 3-generation studies from human populations, I guess, and it's not that easy to get. In Kazakhstan, we had a 3-generation study, and indeed we took into account the spermatogenesis and the duration in that study design. So, but that's mini-satellite mutations, obviously. But important to remember that the mutation rate went up in the children, but then went down again. So back to baseline. So again, how permanent are these changes?
I just have a comment, whether we need to provide recommendations or future considerations, for example, because recommendations is a very strong, right? Yeah. Yeah. Right. Yeah. I think Svetlana mentioned about the time of conception post-exposure, right? I don't know whether there are any animal experiments done in that direction. She mentioned about the time post-conception, post-exposure. Yeah. No, because I think that probably is important in transgenerational effects, because especially for males, because the spermatogenesis process, it's constantly right. So some of the mutant sperms might have been already gone, and then the second generation will come, and the third generation, and how far these effects can be persistent, right?
Are you talking about human studies, or because in mouse studies you have many such, in mouse experiments you have many such studies, because it's easy. Your mate fails in me, and then you expose, or you expose before mating, and you control everything. But I was just now, because you mentioned this question yesterday from group, was it group D, about F0 and F1, and I realized they were confused, and I was also confused. I didn't really, and then I gave an answer, but later on I realized that actually this is not the determination of F0, F1, F2 depends on what you're looking at. and and I think maybe we should explain it to them, because for us, we were focusing on really transgeneration effects, which means preconception exposure. And then for us, F0 was the baby in utero exposure. This was F0. But if you look at effects overall, of course, the mother was F0 and the baby is F1. So, and I think maybe we should explain this, that it's not a fixed, there is no fixed definition. It depends on the endpoint.
It makes the writing easier because you don't have to.
Yeah, right. Yeah. So maybe this you could add this as an explanation, because probably the other groups have also problems with this. And maybe to come back to your suggestion to have clear terminology, this needs to be clearly explained. Yeah, yeah. Yeah, between us, and I think for geneticists, I'm not sure whether they really focus on exposure effects. For them it's clear, okay, here's the father, here's the child, and that's F0, F1. But for us, the moment of exposure is really important. And I think also when it comes to, I'm not sure if people realize this, when it comes to the problem of chronic or acute exposure, and you have chronic exposures in MAIAC with occupational settings, but then the children live in the area as well. So, yeah, it makes it extremely messy. So you need either an atomic bomb or tests. I mean, your veterans, or medical exposure. Yeah, but maybe we should make this also clear that this is. Yeah, right. Yeah.
Or under this chronic exposure scenario, you can do the genetic analysis of mutations from the blood sample of a newborn. Then it's like a without exposure of the adult, and then you can see the mutation rate in utero. Yes, I agree. But at least it's not the organism. You're right. But at least it's as early as you can go.
It's still exposure of that infection.
In that case, you should go through the criteria how to select the study group, keeping all these caveats in mind, because if it could be a mixture of effects, we really want to have a purified group if planning a new study or selecting from the existing population, taking all these.
So then the verdict from this is that, for example, the MAIAC cohort cannot be used for determination of hereditary effects, transgeneration, unless you move them. Yes.
So definitely that's what makes the number of studies smaller.
Yeah.
So there's a large number of step populations you can do.
There's studies that address the question, but then when you look at it, it's environmental exposure, and you cannot be confident of the timing of conception, exposure, and yeah, so you become. It's really limiting to be able to. You end up with a much smaller set of studies that you can be confident that it's definitely preconceptional, and that's the point. Is that.
And then there is always the background radiation anyway. And when I think about the A-bomb testing in Kazakhstan, the dose rate was very high in the beginning, after the tests, and then it went down. It goes down much faster than in nuclear accidents, power plant accidents, for example. So I think the second generation would be very close to background there, if I recall.
So I think what you mentioned, that it's a terminology designed not in relation with exposure. It's just you have a parent with a certain genetic background, and then you look at the F1, F2. Do we recommend something that, because I think the easiest solution is just that everyone, when writing a paper, just defines in their paper what they are using as F0, F1, F2, because it's not immediately obvious. I don't think there is any closed definition that works all the time.
I agree, but it's important to make them realize this, because I think they were looking for a clear definition and it does not exist. So for us, it is in relation to exposure, to the exposure scenario. And Cisco, another thing that should come up again in the discussion is how do we go ahead with writing the review publications? No, we we have to remember this.
Just one more thing, because we briefly mentioned the timing and the relevance for oogenesis and spermatogenesis. Do we want to say anything about that in our? So I've removed the word recommendations. It's now filling the gaps. Do we want to say something about that, or is that just something that goes in the list of minor things?
So for some of the sampling that we put forward for whole genome sequence analysis, we made a limit of 4 months, 4 weeks. We'll have to check that. It might have been 4 months since returning from the last test, to allow for the fact that we wanted the spermatogonium. That's what we were basically 4 months, so that the child that we were sampling was conceived at least 4 months after returning, on the assumption that was the time of their last exposure, so that there is a time gap that you're then sampling the effect of any exposure to the spermatogonium.
But in your follow-up studies, you always concentrated on trio. That means only parents and one child. You're not bothered about the second child.
Yes, and we follow-up studies could look at whole families and you know, the SIBs, but it's basically a practical, you know, it's about scale, and it's about you know, being able to do the study.
Yes. So I think just to catch up on this, the recommendation, if we may give one, like looking at 3 generations, it's a practical one. Of course, it would be fantastic to look, you know, at the future generations, but it's not going to be doable, I think.
It's just impossible. The SIBs, yeah.
Because then you have the time effect.
But it's still a practical thing. It doesn't mean to say that if an interesting family were, you know, you could go back and possibly explore the SIBs, or, you know, so it's just purely practical.
I am not sure if you can say this, Prakash. I would be very careful that you can derive the information about the time effect, because you will be then comparing maybe families with 2 children with 1 child, and this, you know, this makes things complicated, because we don't know what affects having a second child and third child, and this adds a level of complexity.
And we know that.
No, I know, but it may be
Yeah.
useful from the point of view of radiological protection, because these individuals are already exposed, right? What advice you can give them, right?
Yeah, I think that
Address the time gap between exposure and conception to identify. So now it's to fill the knowledge gap, we need to address the time gap between exposure and conception to identify sensitive stages in oogenesis and spermatogenesis, because that was on our list of questions, and I think it is relevant. But given the data that we have now, it's clearly still a knowledge gap, and how we do that.
I think it would be challenging to do in human populations, but and then again, if you are looking into the spermatogonia, and if the mutations are there, they will go on. So DNA damage may disappear, but mutations are persistent. And
the persistence and how long the effect can be transmitted, for example, right? Whether it's immediate or after second conception or third, for example, right? Yeah, something. I think here it's enough just to say it's a gap. Yeah, that's a gap. Yeah. So maybe, if someone is doing an animal experiment in the future, probably that can be considered. Yeah, exactly.
There are, I have been doing this. That's why I have been doing this out now, Sam, with mice when I was working in Essen. Exactly this. We were exposing animals and then mating them different times after exposure to see which stages are more sensitive and more resistant.
I was from the point of view of the knowledge gaps, and again, we discussing the populations that exposed either occupationally or environmentally. Shall we also highlight the cancer patients and offspring born to cancer patients, because it's really the increasing burden of cancer, and this is the important population that also the transgenerational, intergenerational effects could really be a big concern. Of course, we cannot recommend about changing the modalities of treatment. It should be done, but in terms of the health of the offspring, shall we put it into recommendation? Just like, yeah, okay, yeah. Mm-hmm.
Something else we should probably also mention is this: how transferable are results from experimental systems to humans? Because if that was what we were saying in this time gap thing, but I think what you were saying about taking into account cancer patients is yeah.
Does that come under our discussions about future cohorts, potential cohorts? Because there was a few other, so we could, potential future cohorts.
Oh, I see.
Also probably important to the Fukushima population, because they also, it should be concerned about the health of the next generation. I'm not talking about the exposure levels that are definitely in the low dose category, but I think this also should be relevant to the, I don't propose any studies on transgenerational effects in the Fukushima, but I think the findings from the studies of other exposure settings could be relevant and could provide the information to the population of Fukushima. That's what I'm trying to say. Yeah, oh yeah, sure.
I'm just not sure if it's going to work or if it's would be a good study, because there were lots of terminated pregnancies in that population.
I'm not proposed to study, Dimitri. I'm telling that what we are doing now should be relevant and applicable in terms of recommendation and communication.
Oh, okay.
Not launching any study.
Because that's a very peculiar cohort.
Agree. And again, the doses. Yeah.
And they suffered enough from the stigma and.
Really, no, it was not my point to launch any study in this population.
Okay, good.
But it does mean, do we need to include that in terms of education and being able to share information that people can use and trust? Is that something that comes out of this group or another group?
yeah, we can address that to group D, I think.
Yeah, no, you see, I mean, why I was mentioning about the timing, because the recommendation means, for example, women above 35 are not advised to have late pregnancy. That is a recommendation or an advice, right? In clinical settings. And if you know that time post exposure, this is the time period, like 10-day rule in utero exposures. So that's recommendation, right? Something like that. Okay, if you want to have children, wait for 2 years, 3 years after radiotherapy or something like that. No,
We're not in the position. Yeah.
Knowledge gap we have.
I think the only message that we can do in terms of recommendation would be that in the radiation protection system, there is allowance for hereditary effects. You are protected. And scientists are working on that. Scientists and radiation protection community, radiation protection regulators working on that. So you're protected. No, because I don't think we need to. People already are afraid enough, which is evident from what happened in the Fukushima evacuated population.
There is a difference of telling them the effect than communicating that, okay, after 2 years, this is outside of our scope.
Yes, I agree. I agree.
This is a good point to see if there's any final things that we need to put in, and then we can go to the process for the review. So I'll just go and
We need to form the structure of the review, right?
if there's anything important that we haven't discussed.
Yeah, well, in a way, if we can conclude something on the sensitivity of the germ cell stages, then it would be possible to say when it's more safe and less safe, and that kind of things we can say, but perhaps not go to the recommendations to individuals, because it depends very much on the context, what it is about, and I think it's might be more relevant for the pregnancies. And so of course, like early pregnancy, before you know you are pregnant, it's more likely that after radiation exposure, it is either that the embryo will die and there won't be pregnancy, or you will have a healthy child. So these are the two options then, but I don't know if we have too much to say about preconception, except the sperm is more vulnerable than 3 months waiting.
And another point, that if you miss something, right, you will have access to the recording.
Sure.
Yes, but I'm not going to go over all the recordings before 12 o'clock this afternoon. Can you read out what you've got? So for the thing and knowledge,
We share. That's right.
We could, but it probably takes a lot of time to, because I have to get it on a USB stick or email it to you. Well, I could possibly, no. Yeah, or I could just read out what I have. Maybe that's faster now. So I think for filling the knowledge gaps, the things we address is that continue whole genome studies in the trio studies and also in mice, using suitable methods to detect specific radiation effects such as deletions. Pay attention to dosimetry. Consider a variation in dose rate, so acute versus chronic, internal, external, radiation quality. And do this in trio studies, but also in animal studies. I had a typo here. Consider all outcomes, including those with uncertain relevance for detriment, because it might turn out they actually are relevant, and it contributes to general knowledge. For the doubling dose, that requires collection of evidence beyond the discussion that we've had in the past 3 days, so it requires more work. Consider both paternal and maternal exposure, and if feasible, have 3 generations in human studies. Address the time gap between exposure and conception to identify sensitive stages in oogenesis and spermatogenesis. And in addition to occupational and environmental exposure, take into account cancer patients because they are an important group and impact on future generations is important there as well. And transferability from animals to humans is an important issue to address because there are important knowledge gaps there. That's the ones I have.
So it's probably implicit in the whole genome assess, but it's really sort of big data analysis of all of that information and sort of integrate. So it's methods for that, not just generating the sequence and mutation data.
Should I add here that for, particularly for epigenetics, look outside radiation research to establish more knowledge?
For mechanisms, yes, understanding mechanisms.
Have we got anywhere confounders? We had quite a lot of discussion about confounders.
Yeah, that was actually on my other list. Perhaps it should be here as well.
I think so. Yeah. And that's
Background rates.
perhaps we also could recommend for the research that prepare some sort of quality guidelines for the research, what should be taken into account and how, and what the methodological issues. And that could be very good as a paper as well for the reviews. Prakash has kindly agreed to act as guest editor for the special issue. Would you like to say something about how you see the papers?
So I haven't checked with Gail yet, but I think she would encourage to have the special issue, and I'm one of the associate editors. And when Cisco asked me, I accepted to be guest editor for the special issue. This covers all the 4 groups, right? So 4 groups. So what would be the timeline we are looking at by end of the year? Possible, right? Yeah. Okay. So in the meantime, I will check with Gail, and we can launch this special edition of International Journal of Radiation Biology for our workshop.
Yeah, actually, I think that it should be quite easy to prepare the plan, and that's what we need to give to Gail. Gail, the plan of the content, and who will titles of yes.
And I can propose this, and then
right, and for us it would be important to have the titles. Of course, we can discuss this further at the DG meeting today, but we should really engage the experts here because, and now it's fresh in mind, so I would really recommend to start writing quite soon.
At least the structure to start with, how we can process, because we had 22 questions in different topics, and there are overlapping questions as well. Whether we combine everything into a set of topics, or we go with whatever the differentiation we had made, like human studies, experimental, and then mechanisms. That's also possible. Yeah. And then the format, how we go with the review of literature, for example, and then future prospects for each section. I think then we can go with a general summary or future perspectives, for example. Is that okay? And probably we need to decide who will take care of the different sections or not.
So yes, so we've had these special issues after each melody workshop, so there is some routine already. So I would say that there is the guest editor, but the group as of editors, guest editors, yes, and who is dealing with what. They have to supervise some of the processes of papers that are coming up, but the guest editor cannot be involved in the review process of the papers that he or she is involved in. So that's why there has to be a group team. And as for the topics, I think that you would be free to suggest, and then we can, work from that as well, rather than defining exactly how to move on.
I think all of us will be involved.
I think so, yes. Be prepared.
Maybe we can also suggest topics, but maybe I don't know, different approaches, but it worked well in previous special issues that the main responsibles for the individual papers make a very draft structure of the paper, and then contributors say where they want to contribute, because if we start suggesting each of us, then there might be overlaps and topics which are not covered. This is one thing. The other thing I want to ask, what is the timeline you are thinking of? Of course, I know it's very preliminary and then everything can be extended and so on, but just what is the timeline you are thinking of?
I think for the melody workshops, the usual timelines have been that within 1 year it is out already, so it's published. So having manuscripts within 6 months or something like that, then the review process, then publishing. And they make, the papers may appear different times, so I think we should try to go for open access. That's something as well that we need to think how this will be supported, either by the Institute of the Lead Author, or maybe Melody can sponsor something.
For these workshops,
Yes, very good.
it's not necessarily just people that institute has an agreement.
Yes,
Fine, but it is
right.
So what number of papers are you thinking about? So if it's one special issue and there's 4 groups, so at least 4 reviews, but it could be more than 1 review from our group.
Yeah, at least 4 reviews, I would say, according to the groups that we have. But in our case, I think there is so much information that we have to split it further. Yes.
When we were organizing the special issue on the AOP with Nobi, I think his requirement was to get a commitment at least for 6 or 7 papers. So just a comment that I think the more papers the better for the journal, and so I think we're not limited to 1 paper, and if people have other contributions, they will be happy to take.
Just, can I ask a precision question? But it can also be not the only review papers, but original studies as well. So it can be both. Okay.
That would be split to animal and human, but I particularly like your presentation and your review of the human trio studies. But, I mean, that could actually just be broadened out to review of trio studies, whether there's animal in there as well. Yeah, so I can actually see that as a single review. There's a possibility of this, or key messages, summaries from this that maybe get published elsewhere in full, and then it contributes. Some of the review contributes to the structure that you talked about before, epidemiological experimental mechanisms. So bits of whatever review we come up with could inform or could contribute to elements of those. But I do definitely, I mean, I'm probably speaking out of turn because it's your presentation, but I definitely see trio studies as being one paper, one review. But that would be my thing, because then you can lead to future, it can bring it to where the future and maybe the methodological considerations for future trio studies could be one story.
So I understand from Andre's initial comments day before yesterday that melody workshop summary also need to be written, right? No, there is okay. No, the review papers are 4 different groups. So overall summary, there is no need, right? Oh, okay. Editorial. Yeah, editorial, something like that. So that includes. So you have 4 + 1 at least, and then anything other members can contribute. That can be some original, yeah. 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 I think that should make up to at least 8, 9 probably, right? Yeah, and I think it's good to think of potential reviewers as well, because that's very, very important to review the manuscripts, because I have been noticing in IJRB that it's very difficult to find reviewers, and IJRB doesn't allow authors to suggest reviewers. So that's another. So the editor has to find out the potential reviewers.
Just going to throw something out there. IJRB, I'm just thinking of, is that journal fixed? Or, okay. I was just thinking of a broader audience that might be more environmental as well.
Yeah, International Journal of Radiation Biology has been quite often our journal, but we have had once Environment International for the non-cancer effects. And yeah, it's a higher impact journal, more general, and I think there have been more citations to the papers as well, which is interesting. But
when you make this open access, it is available for everyone. It doesn't matter where it is published. Yeah, which journal you prefer, because open access is available to anyone.
Just to complete, we also have the papers from in radiation environmental biophysics. We publish the papers from the workshop on non-homogeneous dose distribution, but we don't in radiation environmental biophysics we don't have special issues. We have theme issues, so it's an regular issue with a couple of publications on a particular topic. But I know that Gail is very much interested in special issues because she thinks this drives the impact factor. So it's easy to collaborate with her, and and that's important.
Just thought I'd throw it out there.
yeah, I think that in fact, International Journal of Radiation Biology impact factor has been going up, so it's not reasonable.
Because of some special issues. Yeah. Or we can decide probably a rough title. Oh, okay. We have 10 minutes. Yeah, we have 10 minutes. Right? The title. Yeah. That is next step. Yeah. You have whiteboard? No, Zoom has whiteboard, you know.
I think it's just a broad theme.
Yeah, all subscribers have whiteboard. Yeah. You see, that can be saved anyway or sent to everyone. So you can't write on your keyboard. Okay, Rona, start.
Well, I've already said I think it would be really nice to have it as a standalone, sort of the future
Okay, so you
trios and future trios study the methodological, challenges and approaches.
But we talk about humans, of course.
Yeah.
That's
All
right,
right.
in humans. Yeah, that's it.
Original. Is that your idea?
Well,
You want
I
to?
don't know, reaching your talk.
Y-
No, I mean, it's basically, I'm suggesting that given the sort of, that as part of the recommendation, that we might want to have a separate review of what we have learned and what we can learn and how to best design trio studies moving forward would be one. No, there's, yeah. Okay.
I mentioned to Rona yesterday that at RSN, Dominic, postdoc Aidana, whom you know, she's been organizing, Aidana Amrenova. Her objective of her postdoc at RSN, she's formally supervised by Dominic, was to do a review of transgenerational effects. And this was before I learned from Rona that you've been working already on a systematic review. And through, I think it's been 5-6 months now that Aidana at RSN, through meetings, we decided that we will attempt actually 3 systematic reviews: transgenerational effects in humans, in laboratory animals, mammals, and then environmental species. Okay, exactly. Yes, this is what I said yesterday. We should meet and discuss and see if we drop this work because you're doing that. At which, what is your protocol, inclusion, exclusion? Is there a chance for us to contribute, or since we're talking about now a special issue, I think that's becoming very relevant to this discussion. That's why I'm saying that. I'm sure Aidana will be also discussing with Dominic, within other groups, and I think we're not in a position to finalize this because I'm speaking for Aidana and Dominic, but maybe they have other ideas. So yes, exactly.
So I think it makes a lot of sense because, provided, so we can go through we can have a separate meeting and go through the approach that we've taken and where the gaps are and how maybe we can work together. But that's the 3 separate sections. And the environmental section is the least worked on because it's the most complicated. It's the most complicated. So we've got lots of papers, but actually to address the question, the issue of timing of exposure is the issue. Yeah. So and then pulling data. But actually, one thing that we also want to be able to comment on is the future design of studies that are designed with some sort of a view to being able to be pulled with other studies moving forward, because there's such variability and heterogeneity between all of the studies. It's been really difficult to pull them together in meta-analysis. So actually, that's a sort of an outcome that we need to be highlighting, that as a field, we don't have a consistency in how we perform these studies that will lend itself to future meta-analyses.
One question regarding the papers and reviews, because we were asking many times about what did we learn since the last report in 2007. So our reviews should go back to 2000 and review of the literature from 2000 onward. Okay, just yeah.
Focusing on the recent information and.
So why don't we propose that there'll be some contribution of the systematic reviews, whether it's combined, whether there's a sort of broad, we'll have a meeting and see if our criteria are similar, or whether it is, it's basically human studies and all outcomes from human studies, then it's all animal studies and all outcomes, and then it's the environmental exposures. That's the 3 sections that we have.
Okay, so I yeah. It is quite, yeah, well, time will show if it should be cut, I don't know. Yeah.
And you seem to have 3 questions.
Okay, so no, it's one question is registered, but we've broken it down into 3 sections.
Okay, so I think that epigenetics should be one of the reviews, and it could cover all areas, also the somatic cells. So what do we know about the epigenetic methods but in humans? And I think biota might be a bit different if they discuss the transgenerational already. But anyway, what are the prospects? What do we know, and what could be perhaps done in the future? And another area is then the experimental models, and to consider the germ cell stages and sensitivity of comparing the species. And it should also include consider the endpoints, context of endpoints. Just some proposals.
So they to be written up here. Yeah. And then maybe include some sort of a review of the evidence as well.
In the first paper, it's human. Because we have trio studies in animals. Other aspects. I just remember the summary. At some point, you suggested to put it in the summary that suggestion and recommendation for future studies to be driven by hypotheses or specific conditions. Knowledge is interpreted. It's not for the paper, but for the summary. The future studies meant to be driven by hypotheses and specific questions.
So the first paper that's about trio studies, would that include a review of the epidemiology? What's not, from classic epidemiology, or do you think that should be a separate? Yeah, but we're talking about preconceptional, whereas the others are not. Yeah.
I think you want to refer to that, say something.
Yeah. So I think the trio studies then could have quite a broad introduction from what is currently understood about from the classic epi, and then moving to trio studies. Yeah. Okay. Yeah. 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 Yeah.
Probably they should be rearranged with humans and then experimental model.
It's not method, is it methods? But is it going to be too general?
Perhaps we should slide the prenatal exposure in there somewhere.
Yeah, otherwise it's too broad.
In our context.
Yes.
Yeah.
Radiation.
Yeah, also for the first one, I think there should be radiation mentioned.
Radiation-induced epigenetic effects.
In the first title, there should be radiation, right?
In the first title, I understood that it should include not only radiation or ionizing radiation, but even secondary exposure to other agents that.
Yes, I think it would be useful.
But we've just been thinking that the context or the introduction should summarize what's been learned from classic epidemiology or epidemiological studies. And so actually then I think it would possibly be too broad if we went out with radiation then. If we just focused on all the epidemiology and then the trio studies for radiation.
I think we would not be too extensive about different exposures, but if we see on the review ionizing radiation, of course we get the priority, but if we see, for example, the chemotherapy in cancer patients or some other factors that play a role, the exposures that we should want to mention, that we will include them, but in a concise form, not
Yes,
too much.
not too much. Yes, we cannot cover everything, but so of course it's a radiation journal, and the topic focus must be in radiation exposure and effects. Same applies, of course, on the epigenetics.
We want the position paper on the symmetry in relation to parental exposure.
We have to consult about it, whether there is enough.
Yeah, or if it's in the incorporated in the kind of quality assurance and I think
He should say.
perhaps these three papers, I think this,
Start with, I think we can go with this.
yeah,
These three and
work on that. So who would take responsibility for the first one?
I'm sorry, can we ask Svetlana? I don't remember if the data she presented on congenital malformations in the Maya workers, if she wants to include this.
Or it's more about the reviews, not about the original research. So I think it would be important to have some original papers included in this special issue. But Svetlana, could you please tell us if the data that you presented on Tuesday about the congenital malformations in the offspring of Mayak workers, were they already published, or you're going to publish, or you would like to include these results as the original paper into the special issue?
These results were published in Journal Health Risk Analysis in 2018. And now we are going to conduct much more larger research, our research.
Do you think it would be feasible to finish the larger analysis by the end of the year to summarize
Maybe.
and update,
Maybe.
or to
Maybe.
prepare paper? Maybe we can include it as.
I hope that we will be able to finish our work about congenital malformation in offspring of Mayak worker cohort to the end of this year. Yes.
Do you agree if we include this paper into the write it on the whiteboard as the proposed paper for the special issue?
Thank you for your purpose, for your suggest. I agree with you. I agree this is well.
Okay.
Thank you. Mhm.
Looks like it's a coffee break there. Just quick comment. I think we should be more specific in titles and say it's trio studies of parental IR exposure, germline mutation studies of irradiation effects in animal experimental models, role of epigenetic mechanisms in inheritance in human. Like we should be more specific what we want, because this is too.
Yeah, no, I think we can do that when we discuss further.
Okay.
At the moment, it's just a draft.
Okay, good.
Yeah.
Just placeholders. And the second paper, is it proposed to the larger group? Because do we have
Later,
epigenetic expertise in here to write this? Like, I don't feel I can contribute much in the, no, sorry, third paper, epigenetics. Well, to some extent, only one aspect of that. And
we can include.
we need a good expert in epigenetics. If Zdenka,
If they are not doing anything on that, then we can.
yes, it's a proposal to the larger group. Okay.
Because we are going to show this to the entire group later on.
And I think because I mean, I'm seeing that we would have information from the reviews, animal studies and the human studies that's actually embedded in some of these, but we could also write down that there's, you know, systematic review somewhere, so that you know, of the question as a whole, so that at least it's considered. And it, obviously, if it becomes published somewhere else, and then we use parts of it to summarize, that's. But at least if we have it in there, because I'm just I can see that it can. What we're doing can be one thing, but actually, it will inform and form part of the introductions and information that are going to these titles. In other words, it's going to be broken up. But I think somewhere we need to have at least considered that there's going to be a full systematic review on the transgeneral effects.
We should break now, right? Yeah. So in the evening, I think when we have the task group meeting, we may give this idea for the special issue, and then later on we can probably email and discuss about how we can go ahead with this, and who would take up. And Andre. Yeah. Other groups as well, if any. No. Yeah, because this is a workshop related. Yeah, related special issue, not just the task group. So should be fine. Yeah. Okay. I think that's good. At least, you know, if the other groups come up with 2 each, then we have a good special issue.